Okay, we will have each instructor make a dilution of enzyme for their particular lab section at the beginning of the lab section. We will give each student group 1 milliliter of this diluted enzyme that the student group will use for the rest of their class period. So we will have the instructor measure 5.5 mils of water into a test tube, then measure 1 milliliter of the undiluted enzyme, and basically have them mix that in their test tube. The instructor will then pipette out 1 milliliter of enzyme into each of 6 different Eppendorf tubes, and we'll give one of these Eppendorf tubes to each student group. The student group will use this enzyme in this state or make some dilutions of that enzyme to use later on during the lab period. Okay, so each student group will have a full strength enzyme tube that the instructor just prepared. They are then going to assemble 2 tubes, one being the positive tube and the other one being the negative tube. The only difference between these tubes is whether the enzyme amylase will be added or not. So into each of these tubes, the student group will add 0.5 mils of pH 7 buffer, 0.5 mils of starch, and water, 0.5 mils of water to the negative tube and 0.4 mils of water to the amylase tube. After adding that, the student group will add 100 microliters of amylase enzyme to the positive tube. Students will then give the tubes a little mix and start the timer. The group will wait for 5 minutes, and after 5 minutes, we'll measure 100 microliters from each tube into a well in a spot plate that contains a drop of iodine, and then we'll see what the results are. If we take a look at the results, if the amylase enzyme was present, the starch still looks yellow, which means that the starch has been degraded. In the minus amylase tube, the starch test worked because we have a nice black color in the solution. In the initial experiments, the students learned that the amylase that we used pretty much instantaneously broke down the starch. We are going to want to test the activity rate of the enzyme, and so we are going to have to dilute the enzyme to a point where we can actually measure the rate of the reaction. So we are going to have students put together 4 successive 10-fold dilutions of the enzyme. In order to make 10-fold dilutions, we have to divide the full volume into 10 parts. 9 of those parts are going to be water, and 1 of those parts is going to be enzyme from the previous enzyme tube. So we are going to have students measure 900 microliters of water into each of the dilution tubes. They can do that at the very beginning. They can then take their P200, set at 100 microliters, and measure 100 microliters of their undiluted enzyme, and then put it into their first 10-fold dilution tube. Students are then going to close the lid, and they're going to basically agitate the tube to get the enzyme mixture to blend in. Students would then take another pipette tip, then they're going to go into the dilution that they just barely made, and they're going to pipette 100 microliters of that solution out and put it into their next enzyme tube. This is a 10-fold dilution of a 10-fold dilution, which is a 100-fold dilution. Students are going to do that 2 more times to make a 1,000-fold dilution, as well as a 10,000-fold dilution of the enzyme. The purpose of this is to try to find the right dilution that will allow students to measure reaction rate. Okay, we learned that the 10-fold dilution of the enzyme still had too much enzyme in it, so we're going to go ahead and put together our next tube. We'll put water in the tube, and the buffer in the tube. Now we still need the starch here. Okay, that's in the tube. Now we're going to try the 100-fold dilution of the enzyme. I need to measure 100 microliters. I'm going to go to my proper enzyme tube, the 100-fold dilution. I will measure out 100 microliters and put it into my tube. Give it a nice quick mix, and then we'll start the timer. Okay, so we have about 10 seconds left. I'm going to get my tube ready. Group member's going to tell me exactly when.
4 seconds.
4 seconds. Okay, I'm going to go.
3, 2, 1.
Okay, I got it taken out. Put it into our. Oh, this time it looks black, which basically means we still have starch in the tube. So we're gonna keep the timer going. We're gonna wait another minute before we take our next sample.
3, 2, 1.
All right, let's put this in. All right, it still looks pretty dark, but I think it looks a little bit lighter. I'm gonna wait one more minute before we take the third sample.
5, 4, 3, 2, 1.
All right. All right. So you can see that it's not quite as dark right now. I'm gonna go and take one more sample after this one, because I think there's still a little bit of a color change that took place. That looks,
Yeah.
maybe it's still a little bit darker, but it's definitely, much of the starch has been degraded already.
One more?
Okay, we'll do one more. Your sample ready? Put that in. Okay, still have a little bit of a change from last time, but there isn't very much of a color change now, which means that most of the starch has been degraded at this time point. That's 1 minute, 2 minute, 3 minute, 4 minute, 5 minutes. So we basically spent 5 minutes working on this, and most of the starch is gone. Okay, we're going to have students put together their own experiment. Half of the student groups are going to test the effect of temperature on enzyme activity, and half the groups are going to test the effect of pH on enzyme activity. I am going to put together some reaction tubes for pH 5 and see how pH 5 affects the enzyme activity. So once again, I am going to measure the ingredients that I need, putting 0.5 mils of water in each tube. I am then going to put in half a mil of pH 5 buffer into each tube. The next thing I'm going to do is I'm going to add enzyme to the plus enzyme tube, because we want to pre-incubate the enzyme at the given pH. So I'll just measure, and again I'm going to the 100-fold dilution, which was the best dilution that we discovered in the previous step. 100 microliters of enzyme will be placed into the plus enzyme tube, and then we will start the timer and wait 5 minutes for this pre-incubation period. We're not going to put the starch into the tube yet, because the pH itself may alter the rate at which starch is broken down. And we really want to test the effect of the pH on enzyme activity. I add the starch to the reaction tubes. I'm going to prepare my 12-well plate with iodine drops. So I've labeled my plate with plus, minus, and different numbers of minutes. So I'll just add 1 drop of iodine to each one of these wells. All right, there's the iodine. And now I'm ready to add the starch. I have 0.5 mils or 500 microliters set on here. I'm going to add starch to each one of my tubes. I'm actually going to add it to the negative first, and then I'll add it to the positive with the enzyme in it second. Okay, I'm now going to give them a quick mix, and then we'll start our timer. And we'll wait for 1 minute before we take our first sample out. Okay, I'm going to start taking samples now. 100 microliters with the enzyme, put it in there. And then, whoops, 100 microliters of my control, put it into its proper well. And what I can see is that both of the reactions turn black, so there's still starch present. Kind of remind ourselves what happened at pH 7. We saw some significant reduction in starch levels around the 3-minute mark. So we're going to keep taking samples at 1-minute intervals. There's the positive. Then we have the negative control. All right, I can see a difference between the positive and the negative already. Looks like the positive with the enzyme in it looks a little bit less black, a little more brown. It's a good interval.
5,
Okay,
4,
I'm going to plus. And do my minus. Okay. Mine still looks nice and dark, so starch seems to be just fine at pH 5. Definitely can see that there is less black in the plus enzyme tube. Plus sample. Minus sample. Okay. Starch does not seem to be affected by pH 5. Sample out. All right. Looks like the enzyme works pretty well at pH 5. One thing we really need to do is compare the pictures, right? I'm going to demonstrate how to put together a temperature experiment. So once again, we're going to put together a positive, which is going to contain the enzyme, and a negative, which does not contain the enzyme. So first of all, we're going to be measuring water into each of these two tubes. We are going to put in some pH 7 buffer to both tubes. Make sure the pH stays steady as the experiment continues. And we are going to put in the enzyme next into the plus tube at 100 microliters. Again, we're going to go to the 100-fold dilution because that's the one that worked the best. Into the plus tube. Do a little bit of a mix. Now we're ready to put the tubes into the ice bath. Here. We're going to let those tubes incubate for 5 minutes before we add the starch and begin taking our readings.
There we go.
All right, so while we're waiting, I am going to put some iodine into my spot plates. Have them plus 1, 2, 3, 4 minutes, minus 1, 2, 3, 4 minutes. I'm actually going to keep going. Have a plus row again at 5, 6, 7, 8 minutes. Have a minus 5 minutes, 6 minutes, 7 minutes, 8 minutes. Okay. All right, so the only thing that we still don't have in our test tubes is the starch. So I'm going to go ahead and measure 500 microliters of starch into each of my tubes. They're over here in the water bath that has ice in it. So I'm going to go ahead and measure the 500 microliters of starch, put that into each of the two tubes, and give the tubes a quick mix. And now we're going to start the timer. So we're going to take our first sample at the 1 minute mark. So I'm ready to take samples out to put into the iodine solution. We're going to do the plus first. Grab that in here. Looks nice and black. And then put in the minus enzyme. Also looks nice and black. All right, wait another minute before we take our next sample. The 2 minute samples. Do the plus first. Location. Next sample, minus sample. So that in. Both look black. Okay, I'm gonna start taking my next sample.
5.
Plus enzyme first. Minus enzyme next. Got a piece of ice there. That's the 4 minute mark. minus sample. All right. Minus enzyme right here.